Pathogenic trials operate on a precise protocol. A single controlled substance is administered to healthy human volunteers to map its specific effects on the body. Biological reality complicates this premise. No two participants share the same genetic disposition, constitution, or baseline vitality. Because of this diversity, the same substance produces different symptom sequences across the cohort. These diagrams show how one medicine triggers varied biological paths. Variation exists within the individual prover too. As doses repeat and time elapses, symptom expression shifts, requiring constant monitoring. These outcomes are expected within the framework of aphorisms 113 to 115. They represent the diverse ways the human system processes a medicinal stimulus. Objective methodology exists to decode these variations rather than to suppress them. In this framework, biological diversity provides the raw data necessary for a complete drug picture. Outside observers often expect trial data to be perfectly uniform, assuming that consistency equals validity. Hahnemann's directive explicitly counters this. Researchers are forbidden from forcing a prover's subjective experience into a standardized pattern just to make the data look cleaner. The operational requirement is a time-ordered symptom diary. This document captures the exact sequence and duration of every emergent state. Aggregating these differences into a generic average destroys the data. Knowing if a symptom belongs to a primary or secondary phase of drug action is essential for clinical accuracy. The goal is to capture the broad spectrum of a drug's power by embracing individual nuances across a diverse group of provers. To extract the true medicinal signal from biological noise, researchers apply rigorous symptom filtration and maintain absolute pharmaceutical traceability. We move from the human volunteers to the data points they generate. A common symptom is an effect reported by a large proportion of the group: generic dull headaches, fatigue, or mild nausea. These prove the substance is biologically active, but they offer low diagnostic utility. They are too generic to differentiate one medicine from another. The focus instead is the characteristic symptom. This is a peculiar, uncommon complex with high individualizing value. Frequency describes occurrence, but characteristic specificity guides remedy selection. The funnel separates broad biological action from unique medicinal signals. This raw data must meet 4 pillars: location, sensation, modalities that change the symptom, and concomitants that accompany it. Supervisors use strictly neutral, non-leading questions to extract these pillars. This prevents expectancy bias from polluting the prover's genuine experience. This complete symptom matrix transforms an ambiguous human feeling into a highly specific clinical coordinate. Integrative medicine often faces the temptation to use chemical analogs or related plants to fill missing symptom data. The no surrogates principle in aphorisms 118 through 120 strictly forbids this. Chemical similarity is insufficient. Identical formulas can produce vastly different dynamic responses in the human body. Botanical relationships do not replace trials. Even plants in the same family, as shown in these historical engravings, exhibit contrasting mental and physical modalities. Preclinical animal toxicology also falls short. Animals cannot articulate subjective sensations, emotional states, or environmental modalities. We must also account for extreme data, or idiosyncrasy, described in aphorism 117. This is a marked physiological alteration resulting from a minute exposure that most provers tolerate without reaction. An idiosyncratic response reflects individual susceptibility. It is not automatic proof that the symptom belongs to the universal drug picture. Researchers verify these reactions by ruling out external triggers and establishing a recurring pattern within the individual. Pure pathogenic knowledge requires human trials. You cannot substitute an animal for a human or accept a hypersensitive reaction without disciplined verification. Scientific validity depends on the physical and administrative governance of the clinical trial. The proving dose requires careful calibration. It must be strong enough to elicit discernible symptoms while remaining below the threshold of toxic harm. Excessive exposure produces violent, nonspecific toxic reactions. These mask the subtle modalities researchers need to document. Aphorisms 122 and 123 mandate traceability. An uncertain substance produces uncertain knowledge. Without authentication, data is void. Documentation requires material identity, preparation records, batch numbers, and strict chain of custody logs. Allocation concealment, or double blinding, is an absolute requirement. Blinding removes expectancy bias from the prover and suggestive influence from the interviewer, protecting the integrity of every observation. To safeguard participants, the protocol defines stopping rules. Severe adverse reactions, or clinical red flags, trigger an immediate cessation of dosing. The medical lead then uses a controlled unblinding pathway to manage the volunteer's safety. A robust protocol provides the physical framework for the entire study. It secures the safety of the volunteer while anchoring the trial's scientific validity. These disparate methodologies create the life cycle of a validated data point. A raw symptom emerges from the variable human baseline. It survives idiosyncratic verification and is filtered through the 4 pillars. This data point remains anchored to clinical reality through its batch traceability and blinding logs. A Materia Medica entry is only as reliable as the objective protocol that generated it. Integrating biological diversity with pharmaceutical accountability produces a clinical tool that is both human and scientifically defensible.